Welcome, everyone. It's my pleasure to introduce Dr. Gustavo Angarita from Yale University and Department of Psychiatry. It's interesting talking to Gustavo today, how we both came into using PET to understand molecular basis of addiction. Gustavo completed medical school in his native Colombia, then came and completed a psychiatry residency at Yale. And then did an addictions fellowship, 2-year fellowship at the Harvard hospitals, and came back, and Yale smartly hired them onto his faculty, where he was mentored by, as I just inquired, the Bob Mallison, who was a former colleague of ours. Bob unexpectedly passed away a couple of years ago, but was a real driving engine in the original PACE. And so Gustavo of late has been very successful from the NIH standpoint, getting very high scores on a grant, and we're excited to hear him today talk about how he's using PET to inform our understanding of cocaine use disorder. So thank you, Gustavo, for coming, and I'll leave it to you.
Thank you. Thank you so much, everyone, for the invitation, and also Jake for the introduction, and for everything yesterday and today. Yeah, this is a study, or a combination of studies that we have exploring synaptic density in cocaine use disorder. And the goal is to see to what extent this can inform future medication development efforts we can try when it comes to pharmacotherapies. And I have no financial disclosures. This is a brief outline of the presentation. So we have some background in terms of cocaine use disorder, and the preclinical studies on aberrant synaptic density that preceded the clinical work that we are doing now. And then the process for us to get to the point of imaging synaptic density in humans now. Then I will be talking about a cross-sectional study that we did first, then a longitudinal study that we are currently doing, and then I'll be briefly talking about potential future directions as it pertains to medication development efforts. So in terms of the background and cocaine use disorder, in some of our conversation this morning, we were talking how cocaine use disorder doesn't have the impact that opioids have when it comes to being categorized as an opioid epidemic. But cocaine use disorder is an endemic problem that is highly prevalent. We have at least 2.2 million persons who use cocaine on a regular basis in the US. We have more than 1 million persons with cocaine use disorder in the US. It is a very common cause of lethal overdoses, independent of cocaine samples being contaminated with opioids. And it is a common cause of visits to the emergency room, more than 500,000 visits per year. And every time, at least in New Haven, we go to the emergency room to work there, there is one patient who is under some sort of observation in the psychiatry unit in the context of cocaine use. In spite of all of this, we don't have any FDA-approved pharmacological agent for the treatment of cocaine. So this slide shows the natural history, the progression of, in general, substance use disorders, but also cocaine. We see different dimensions in a person's life. We have the family, occupation, work, friends. If they initiate using cocaine, we see how that occupies a little piece of the dimensions of that person. But as they go from initial use to intermittent use and then frequent regular use, we see how the drug use impinges on all the other categories of the person's life, leading to multiple biopsychosocial problems at the medical level, cardiovascular problems, strokes, or psychiatric difficulties, mania, psychosis, housing insecurity, and legal problems. As someone goes through the progression of the natural history of the cocaine use disorder, they engage in the different stages of addiction. So they have binge use, intoxication with cocaine, and withdrawal, negative affect, and then preoccupation and anticipation about the cocaine use. All the behaviors around these stages of addiction are very hardwired and very recalcitrant to treatment. And we don't know very well what is underneath these behaviors and whether neuroadaptations at the level of synaptic density is what is driving this. So seminal studies at the preclinical level in the 1990s explore what were the effects of administering a vehicle, amphetamines, or cocaine to rodents. And so they examined the dendritic branches, and they also examined the spines count after administration of amphetamines, cocaine, and vehicle. They did this in rodents 1 month after the last administration of cocaine, and they saw that for both the dendritic branches and the spines count, they saw an increase in numbers in the nucleus accumbens and in apical and basilar dendrites of the medial prefrontal cortex.
I think you may be able to grab that little line there and move it. The green, red line with your, you may be able to grab it and move it. There you
Yeah. Then subsequent studies replicated this, and the studies replicated this not only after the investigators were administering the cocaine or the amphetamines to the rodents, but also when the rodents were self-administering the cocaine or the amphetamines. The studies also replicated this at different time frames of abstinence from cocaine. So not only 1 month later, but 2 weeks later, for example, 1 week later. They did this also using more modern methods like 3D morphometric analysis. And they also learned what were the molecular substrates that were underneath the microarchitectural changes. At the same time that they replicated all of these results, the different investigators saw significant complexity in terms of the findings. For instance, they saw different effects depending on different subregions of the prefrontal cortex. So they saw increased branches and a number of spines in the medial prefrontal cortex, but in the orbital prefrontal cortex, they saw decrease. So different changes depending on the subregion, opposite effects of opioids and morphine in comparison to the effects of cocaine. And they started to inquire whether these changes were causal or compensatory. So the chicken or the egg question, like what was happening first, if the synaptic density changes or the cocaine administration. Now, at a clinical level, we have very limited tools to know what is happening in terms of the microarchitecture. There were a couple of studies post-mortem using microarrays to examine more than 39,000 transcripts in the brains of persons who had opioid use and in the brains of persons who had cocaine use. And they look at multiple transcripts, including several transcripts from the presynaptic machinery proteins. For example, synaptophysin, synapsin, and synaptic vesicle glycoprotein 2A. In these studies, they saw opposite effects of opioids in comparison to cocaine. So they saw, for example, that in the brains of participants who had opioid use, they saw lower expression of the transcripts for the SV2A, and in the brains of persons who had cocaine use, they had the opposite effect in a direction that matched the preclinical results.
Sorry, how long did they see those changes? A month after?
So they had the brains collected like within, like soon after death.
Oh, these are all the post-mortem.
Yeah.
So, all right.
Yeah.
Sorry.
So this is all post-mortem. And so we had clinically access to the post-mortem studies. Then, I mean, we know that we have access to the structural MRI, and at the level of the structural MRI, there have been several papers showing reduction in the gray matter volume after exposure to cocaine in similar regions of the preclinical studies, the prefrontal cortical structures. Also, some of those studies show correlations in which the longer the lifetime years of cocaine use, the lower the gray matter volume. but there are some mixed results in this literature. For instance, this paper in 2017 showed that when the cocaine participants were compared with the healthy controls at baseline, there were no differences. But after 6 months of cocaine abstinence, in comparison to the healthy controls being scanned 6 months later, the cocaine participants actually had higher gray matter volume than the healthy controls.
Do you know if you corrected for any alcohol use
I don't know if they did that on this, on this study.
in the control group?
That's-
Yeah.
It'd be affected.
Yes.
If it's not one thing, it's another.
Yeah.
Well, alcohol clearly reduces gray matter.
Yeah.
So at a clinical level, we had very limited access to tools. Like we had the postmortem studies, we have the MRI, but the MRI is not telling us exactly what is happening at the level of synaptic density. Any changes on the gray matter volume could be a reflection of different size of the nuclei of the cells or different number of glia cells, or it could be changes in the actual number of synaptic density. So going to the studies that allow us to examine synaptic density in humans. So here we have presynaptic axon, we have the vesicles, we have at the synaptic junction, here we have the postsynaptic cell. So if we zoom in into a vesicle, we see all of those transmembrane proteins and how some of those transmembrane proteins are the classic markers of synaptic density, like synaptotacmin, synaptophysin. Well, the synaptic vesicle glycoprotein is a transmembrane vesicle protein that is, I would say, in quotations, ubiquitous, because there are some papers sort of suggesting that it's nearly ubiquitous, but it is ubiquitous in the gray matter. And because of that, it is suitable for the imaging of synaptic density. In terms of the function, it is important for the exocytosis of the vesicles, for the neurotransmitter release. And it's also important in epilepsy, as mice lacking this protein had significant severe seizures. In terms of the development of the PET ligand, so this was a collaboration between a pharmaceutical company in Belgium and Yale University, trying to look at different and better anti-epileptic medications than the ones they had available, such as the levetiracetam. So they were testing different molecules, and as they were doing the studies on like occupancy, they realized that this molecule could be used not only as a molecule to test the occupancy and the displacement induced by the other medications, but this could be a molecule that could be a PET ligand useful to examine synaptic density by itself. In terms of the preclinical validation studies, so those studies were done in baboons. Here we see MRI and PET summation images. We see the nice binding in the cortical ribbon and the gray matter in the subcortical structures. Here we see time activity curves in which we see high penetration in the brain and fast, rapid kinetics, allowing studies to be done in like less than 2 hours. We see low binding in white matter, which is also good. So we have low binding in areas that are not supposed to be areas with synaptic density. This is a Western blot. So we see examination of SB2A in comparison to other presynaptic classic markers like synaptophysin. So we see how in different areas, such as, let's say, number 4 is the frontal cortex, we see a good presence of antibodies for the proteins for the SB2A as well as synaptophysin. If we look at number 5, which is the centrum semivale, we don't see presence of neither one of those proteins in the centrum semivale. Here we
Can you go back? This is all C11?
yeah.
Yeah. I was curious because there's all the fluorines there. I wasn't curious how hard it would be to.
That's the latest and the greatest.
Yeah.
one.
Sorry.
This was the first paper.
Yeah. And And then here we see a correlation between the Western blot for the SB2A and the PET signal for the SB2A. So we see a linear correlation between the Western blot and the PET. This is a Western blot for the SB2A correlated with the Western blot for the synaptophysin. So we see also a linear correlation between those two. This is a low power confocal microscopy and high power confocal microscopy. So we see in green the staining for the synaptophysin, in red the SB2A. This white dotted line is the separation between gray matter and white matter. So we see how both synaptophysin and SB2A merge in the gray matter. We can see this at high power confocal microscopy here. And we see in purple the nuclear cell, the nuclei of the cells.
You know, as the group done knockdowns of SB2A to know if that drops the amount of signal present, like in cells, like cell lines that have SB2A naturally expressed.
If they do the knockout, if they see changes in the expression,
Yeah. Or
I
just
haven't
like
seen.
uptake of the radiotracer in the knockdown cell line.
we haven't seen those
Okay.
those studies. I know there is a recent study that is in preprint, and that's the study that is sort of questioning how ubiquitous is
Yeah.
the SB2A and how well it colocalizes with other markers, and whether
Neurons.
and depending on whether it is equally present in like excitatory versus inhibitory neurons. And so in terms of the validation studies in humans, so this is a study in healthy controls. We see again similar pictures to the ones in baboons, similar kinetics in terms of the penetration to the brain and the rapid clearance from the brain. This is a validation in epilepsy and temporal lobe sclerosis. So we have sclerosis of one side of the brain in comparison to the other one. We'll be expecting an asymmetry in the signal between one side versus the other side. So when they compare that in persons with epilepsy versus healthy controls, they saw higher asymmetry index in the person with epilepsy versus the controls in areas such as the hippocampus and amygdala, sort of mapping to anatomical areas with the sclerosis.
Was there also an overall reduction in synaptic density?
Well, you mean in the whole brain?
Yeah.
I don't know if there's a whole brain. It
Or
was
maybe-
more like lateralizing one versus the other.
Other and beyond the validation studies, then the ligand was started to be used across multiple conditions. So they did studies in Alzheimer's disease, major depression. In Alzheimer's, they saw decreased binding in the hippocampus in comparison to healthy controls. In major depression, they saw lower binding in the dorsolateral prefrontal cortex in comparison to healthy controls. And in depression, the lower the binding, the higher the severity of depression. In terms of, PET in a nutshell, this is not necessarily that important for this audience, but so we have positron emission tomography. We have the positively charged electrons that are emitted from the radiopharmaceutical. This collides with the negatively charged electron. The collision from these two emits gamma rays in opposite directions, and those gamma rays are detected by different sensors in the scanner. So for the PET, we have the cyclotron that produces different isotopes. In the case of this study, the carbon-11, we have the radiopharmaceutical laboratory in which we sort of put together the carbon-11 with the pharmaceutical. The ligand is injected into the participant, and then the images are reconstructed in 4D, having time as one of the dimensions. In terms of the study design for the cross-sectional initial study that we did, so we had 15 participants with cocaine use disorder matched with 15 healthy controls. The participants with cocaine use disorder were studied as inpatient at the Clinical Neuroscience Research Unit after 2 weeks of abstinence. The healthy controls were studied as outpatient. The regions of interest were regions mapping the preclinical studies, so different subregions of the medial prefrontal cortex, orbitofrontal cortex, and the ventral striatum. The PET scan took place in the high-resolution research tomography, MRI in a 3 Tesla, and the primary outcomes were the gray matter volume, volume distribution, and the free fraction corrected volume distribution. So our aim was to examine synaptic density in the brains of humans with cocaine use disorder in comparison to healthy controls. And our hypothesis, thinking about the preclinical results, we were hypothesizing higher BT and BTFP in the ventral striatum and medial prefrontal cortex in cocaine versus healthy controls, and lower BT and BTFP in the orbitofrontal cortex in cocaine in comparison to healthy controls. So this will be a hypothesis matching the preclinical results. The inclusion criteria, we had participants from 21 to 55 years old, all healthy physically and psychologically, no significant comorbid psychiatric conditions, no significant other substance use disorders. They needed to have a positive urine tox upon screening, and they had approximately 2 weeks of abstinence from cocaine at the point of the PET scans. The healthy controls had the same inclusion criteria, with the exception of the cocaine use. For the exclusion criteria, basically having any significant dependence on other drugs, with the exception of nicotine, having any other significant psychiatric conditions, or any unstable medical neurological problems, using any psychotropic or potentially psychoactive medications that could affect the outcomes, contraindications to the MRI, any pregnancy or breastfeeding, and abnormal PT and PTT that could preclude the arterial line.
This was a recovery population. They were in recovery.
I mean, they were using,
They
Yeah.
were using cocaine.
they were using cocaine.
Only cocaine.
Yes, they were using cocaine, but contrary to what we see with the studies in opioid disorder, it was easier for us to see a population that was mainly just a reflection of cocaine use and not other significant psychiatric conditions or, yeah. They could drink alcohol or use cigarettes or cannabis, but they didn't meet significant
As long as
substance
they
use
didn't meet
disorders.
that,
Yes,
the criteria for
Yeah.
dependence.
Yeah.
So if you had a positive UDS, as long as they didn't.
That
meet
was
those,
okay.
It was
Yeah.
fine.
So these were the a priori candidate regions of interest. So we have subregions in the medial prefrontal cortex, such as the anterior cingulate cortex, dorsomedial and ventromedial prefrontal cortex. We had subregions of the orbitofrontal cortex, such as the lateral and medial orbitofrontal cortex, the ventral striatum. They were taken from the AAL atlas, with the exception of the ventral striatum, which was manually drawn. So these are the outcomes. We have the gray matter volume using the MRI. I mentioned the AAL atlas. The PET outcomes were the volume distribution, which is the binding concentration of the ligand in the tissue over arterial sample, and the free fraction corrected. These are the results in terms of demographics. So the participants were matched, like the cocaine group with the healthy control were matched by age, sex, and race. They were different in terms of years of education, alcohol use, and tobacco use. Like none of the healthy controls were smoking cigarettes. And we saw no difference in terms of the injected dose of the ligand or the free fraction of the ligand. In terms of the gray matter volume, we didn't see any differences between the groups, so no changes there. In terms of the volume distribution, we saw no statistical significance between the groups. So we saw like slightly lower numbers in the cocaine group than the healthy controls, but none of those reached statistical significance. In terms of the BTFP, we saw lower BTFP in the cocaine group, in the anterocingulate cortex, dorsomedial prefrontal cortex, and anterocingulate cortex, ventromedial prefrontal cortex, and medial orbitofrontal cortex. So the difference between the BT and the BTFP is that when we took into account the free fraction of the ligand, the variance of one group was reduced significantly. So by having a lower variance in one group, statistically,
I assume that was the healthy
we
controls.
yeah,
Had less variance.
that allow us to separate
Yeah.
the groups.
The free fraction is measured from blood.
Yeah, and so from the arterial samples, and it was examined with ultrafiltration.
And what's the origin of that difference?
In terms of, no, the free fraction was similar between the groups. Yeah, so it was that the one group had less variability than the other group. So that changed the actual variance of the BTFP.
And these were normalized to individual FP, so it wasn't a group across the group, right? So for
Yeah.
one subject, they used their individual
Yeah.
free fraction
Yeah,
to get.
each participant had, yeah. This is the visual representation of the results. So each dot is one participant with cocaine in red or healthy controls in blue. And so we saw the difference in the anterocingulate cortex, ventromedial prefrontal cortex, and medial orbitofrontal cortex. We saw no differences in the centrum semivalley, which is the control region, and the area just mainly rich in white matter. So that was expected.
But you didn't see the increases in the ventral striatum, which is what you expected. Right?
Correct. We saw
From-
results very different from what we expected.
Right.
Like we saw the opposite. Yeah,
Yeah.
because
So reductions across the board. And
Yes.
Because you were thinking they were going to be
Yes.
paired, like down in one place and up in
yes,
another.
we were thinking that we were going to see higher synaptic density in the medial prefrontal cortex, lower in the orbital prefrontal cortex,
Just like-
higher in the ventral striatum. We saw just reduction in the medial prefrontal cortex and medial orbitofrontal cortex, and no changes in the ventral striatum. So instead of seeing higher levels, we saw lower.
Was it also chronic use of cocaine, or was it more?
I mean, chronic in terms of preclinical studies, like they were administered for like several weeks or, yeah.
Given those baseline differences in education,
Yeah,
alcohol,
yeah,
did you?
yeah, we examined that, and that didn't change the results in terms of after covariant for educations or alcohol use, the results remain the same. Yeah. And the FP, as I mentioned before, was the same between the two groups.
Did you correlate anything with chronicity or frequency?
Yeah, so those are very interesting and counterintuitive results. Yeah, and these are those results. So we're gonna start with the gray matter volume. So in terms of the gray matter volume, I mean, we saw correlations that were intuitive and similar to some of the papers in the literature. So the higher the lifetime years of cocaine use, the lower the synaptic density in the anterocingulate cortex and dorsomedial prefrontal cortex, and also the ventral striatum. We didn't see that for the synaptic density. And in terms of the frequency of cocaine use, measured by number of days of cocaine use per month, we saw a positive correlation. So the higher the frequency of cocaine use, the higher the synaptic density. And this is one correlation that I consider counterintuitive, because if the cocaine group has lower synaptic density than the healthy controls, how come the higher the frequency of the cocaine use per month, the higher the synaptic density?
So for the areas where you expected it, like the ventral striatum.
So that's one correlation. We also saw that the participants who were scanned farther down in the hospital-based abstinence, like 21 days versus 10 days, or 20 days versus 13 days, they had lower synaptic density. So there was a negative correlation between hospital abstinence and findings in synaptic density. So as they were getting abstinence in the first weeks of hospitalization, the synaptic density wasn't going up but it's actually going down.
So you're expecting an increase
Correct,
based on the COOP study.
based on the preclinical studies. Yeah.
Can you go back to that slide? I'm gonna ask some, probably some dumb questions. Go back to the COOP study. Been a while since I've thought about this stuff.
Were they also absent for a long time? The animals?
Like 1 month, 2 weeks. Yeah.
Let's look at the pictures of the neurons. So you're seeing an increase in spine density. And that's along the axon or the dendrites?
That's in this case, yeah, that's one of the big differences is that the preclinical studies are looking at the dendrites.
Yeah.
The clinicals were looking at the presynaptic part. Yeah.
Yeah. And the spines are supposed to represent areas of release zones from the dendrites, or are they receiving synaptic input?
Yes,
It's
input, yeah.
So if you're seeing an increase in spine density, that would be consistent with a reduction in synaptic input, which is what you're seeing in your PET studies. Right?
Mhm.
So the results may be consistent with this. That's what I think,
Mhm.
right? Does
Yeah,
that
it
make
makes sense. Yeah, because the spines are looking for a signal.
Yeah. Yeah.
And there's
those are
nothing, they
postsynaptic
don't hear
neurons.
anything, so they're looking,
So I think your PET data may be consistent with this, not inconsistent.
Mhm.
But is it also consistent with the increase in synaptic density and frequency of use? Not really.
Well, it depends on what you're looking at, though, right? And where are those regions where you saw the increase?
Almost everywhere, right? It's
Yeah.
Positive
Wait,
correlation.
Go
with
back to,
use.
now where did, I must have missed, so I was focused on the
Yeah.
decrease, which I thought was consistent. Now, where are you seeing the increases and under what conditions? Whether was that absence?
Use.
This one.
So those
this,
cocaine
yeah.
users who use more have higher synaptic density.
Use more frequent.
Yeah, the more frequent cocaine use they had.
It's a stimulant.
Yeah, but if it's decreasing. It's decreasing. Dendritic. No, it's increasing dendritic spines, then the presynaptic should be increased.
Yeah, the way we were interpreting is that each spine should be an area of electrochemical sort of contact. So
Where's the increase? I don't understand this data.
Positive regression.
yeah,
frequency.
so this one, the higher the frequency of cocaine use per month,
Yeah.
the higher the synaptic density in the anterior cingulate cortex, dorsomedial and lateral frontal cortex, and the ventromedial prefrontal cortex.
I would argue in favor of lower dendritic spine density,
Mhm.
postsynaptic. I mean, if that
Yeah.
model.
Yeah.
That Bob suggested holds.
You know, and initially we were seeing this as counterintuitive because we're seeing like each spine as an area of electrochemical contact. So yeah, but then, so we saw those counterintuitive correlations. This is sort of the visual representation of those three, using the anterior cingulate cortex as an example. So higher frequency of cocaine use, higher synaptic density, longer days of abstinence while in the hospital, lower synaptic density, and longer lifetime years of cocaine use, lower gray matter volume. So this is the summary of the results. No differences for the BT, lower BTFP in the anterior cingulate cortex, ventromedial prefrontal cortex, and medial frontal cortex. No differences in the ventral striatum. No changes in the gray matter volume. No specific, no changes in the central semivalley. Positive association between BTFP and frequency of cocaine use. Negative association between BTFP and abstinence.
But those positive and negative are only in the certain circuits. That's not.
And it's
Full
just one snapshot, yeah, so just one cross-sectional scan in some
In
areas.
some areas,
Yeah.
right?
Yeah, just in
But what-
some.
I mean, I think what, no, I don't want to take away from, because
Yeah.
it's an interesting discussion with Rob
Yeah.
and Hank we're talking about. But I mean, we're talking about local circuits and how they're behaving, but you're basically putting this stimulant throughout a very positive stimulant
Yeah.
throughout the entire brain.
Yeah,
So I would suggest also the overall tone of what is actually
Yeah.
going on the entire brain
Yes.
could be different. And these are just
Yeah.
small reflections that show what, the ultimate behavior,
yeah,
the reinforcement behavior.
Yeah.
But that doesn't mean there's differences in plasticity and how the neurons are communicating throughout the brain.
yeah,
The last two findings
Mhm.
seem consonant with one.
Yeah.
And the more frequency, the greater the VT or
Yeah.
LVP. And when with greater abstinence, it's a negative association.
Yeah.
Does that make sense?
yeah, and so that led to like I will show you this comparison between the preclinical and the clinical studies, and then I can show you the longitudinal study that we are doing, trying to make sense of those correlations. So in the preclinical studies, higher in the middle prefrontal cortex, lower in the middle prefrontal cortex in the clinical studies,
Yes.
higher,
Sorry, that's self-administration preclinical studies.
yeah,
Okay.
and both. I mean, in the
Right,
preclinical, it was
right.
the same direction. Yeah. Lower in the orbital prefrontal cortex preclinically, lower clinically, higher in the ventral striatum preclinically, no changes clinically. So we actually came up with this model. So our model was, and this is going back to the background description about compensatory or causal, like which one is first. So we thought that one hypothesis is that before the cocaine use, these persons could have lower synaptic density. And the cocaine use is, in quotations, a self-medication attempt at normalizing that deficit in synaptic density. So the cocaine use in humans may be doing the same as it is doing in the preclinical models. And that's why, so they have lower synaptic density before engaging in the cocaine use. Then that's why the higher the cocaine use, like the higher the frequency of the cocaine use, that is an effort to normalize the synaptic density, but it doesn't reach normality. And that's why the cross-sectional scan still shows that the cocaine group had lower values than the healthy controls. Then they stopped using the cocaine, and then the effects that the cocaine initially had are subsiding. And that's why we saw lower synaptic density with time of abstinence in the hospital. This is not that different from other work in terms of the glucose consumption in cocaine use. The first 2 days, 3 days after cocaine use, we see more glucose consumption than weeks later. So thinking about future directions, we wanted to replicate this with a larger study, examining this longitudinally, looking at other variables such as neurocognition, and doing the studies in opioid use disorder. So this is the longitudinal study that we have now. So the participants with cocaine use disorder are admitted to the inpatient neuroscience research unit. They do an initial PET scan within 48 hours of their last cocaine use with cognitive testing. They stay on the inpatient unit for 3 weeks, and then they do a second PET imaging scan. All of these while monitor inpatient on the unit. Also, they do a cognitive testing after the second PET scan. Then they are follow up as outpatient twice a week to get a sense of association between what we see with the PET scan and time to relapse. The healthy controls do the study as outpatient, single scan with the cognitive testing. Inclusion criteria are very similar, only with the exception that we are allowing a wider range in terms of age. Exclusion criteria also very similar. These are the demographics of participants only in the cocaine group as of June of this year who had done both PET scans, both with arterial line. So the participants recruited is a much larger number. But this is only looking at the longitudinal aim. Cocaine participants who had both PET scans, both with arterial lines. So the demographics are similar. We had a very low number of females in the study, which is one limitation that we have. And this is the within CUD comparison, first scan versus the second scan. So we actually see that the second scan, they had lower synaptic density than the first scan. So this is matching the model that we had. So we see for the ventromedial prefrontal cortex, dorsomedial prefrontal cortex, lateral bit of frontal cortex, and medial bit of frontal cortex, we see lower synaptic density on the second scan versus the first scan. We are seeing that for the BTFP. This is the visual representation of those results. We are seeing the same for the BT. So with the volume distribution, we see the same, like lower binding in the same regions, ventromedial prefrontal cortex, dorsomedial, lateral bit of frontal cortex, and medial bit of frontal cortex. This is the visual
Where is the
representation.
time range?
3 weeks.
2 weeks?
3.
And where would a non-user be on that scale?
Yeah, exactly.
Because
How
there's
does-
a 7% decrease in 3
Yeah.
weeks. You would think that normal would be.
Does your baseline cocaine group, how does it
Yeah,
compare
what's
to the
that retest
control?
variability?
Yeah, so that's a great question. I think it is not far from the, like, for the BTFP, it is like 6 to 7%. For the BT, is like 3 to 4%. So I think that one question is going to be whether in like part of the results are related with the test retest, but and we'll have to see if the results stay when we look at all the participants.
Are you
Yeah.
gonna scan the healthy controls twice?
Only once.
It
Yeah.
looks like it's the abstinence of cocaine that is causing a decrease in synaptic density, right? For
Yeah,
both these
so we are
studies.
seeing that, 2 weeks later, and but yes, one important question is how much of this is related with just test retest variability, and I mean, it could be just by chance that in the test retest variability we see lower in the second scan, or yeah.
But I
Yeah.
wouldn't expect there to be a whole lot of normal changes in weeks, right? Even with usage,
I mean,
like density change,
we,
we don't know.
you know, we didn't think there was 40, 50 years ago, we didn't think there was plasticity, right? Like, and now in terms of how you're finding it in circuits more and more,
Uh-huh.
and it's change over time. And so if the changes have this, it actually makes sense. You know, the other thing I was thinking, just because Bob and Sharon and Gustavo and I had lunch, was Bob was talking about his study on intrinsic variability in the non-human primate, the dopamine study they did a long time ago. And if you have a population that has intrinsic variability that may have an underdeveloped reward circuit, and the way they get that circuit to function better is with the chemical, that could make a lot of sense. The other thing I was thinking about, just in terms of that, is how many times these people this is not their first time going abstinent.
Yeah.
They've had this disease for a long time.
Yeah.
And so how, that number of times and how impaired or how many times they've tried to potentiate their baseline with
Mhm.
chemical use. I wonder how that, plays into how much elevated it is
Yeah.
compared to what its baseline normal station is.
And Mark, in the D2 study we did years ago, I was talking about at lunch. And these were mostly self-administering cocaine.
Nader at all?
This one, yeah, the Nader et al. One, yeah.
There was an increase in dopamine 2 receptor in the dominant monkeys, I think. They were
Yeah.
individually housed.
Yeah, that's a Morgan et al.
Oh, that's the more.
That's the Morgan. The Nader et al. Paper was a longitudinal study. Because we saw that the D2 receptor, D2, D3 receptor started to downregulate. After about a week, and
Mhm.
after a month, they basically reached almost as low as they were gonna go, even if you carry them out for a year. And then when you pull them into abstinence, they start to recover after a
Mhm.
week, and they recover, if they recover at all, it's within a month. And those that stayed down at a month never recovered.
Right.
If I remember that, I mean, that study.
So I guess the healthy
2003.
control data on here would be really helpful, right? Was
Yeah.
it above, is it near the blue line or is it near the?
Yeah.
So these changes can occur
On that scale.
Well, if you, I mean, if it's on the HRT, you should have plenty of controls anyways,
Yeah,
right?
With
Because
this
that,
ligand.
yeah,
But that they didn't scan twice, I guess.
right.
yeah, I don't think. In terms of the scan twice, I don't think.
Has anyone done test retest with UCBJ? That's the question. I thought that was in the first.
Yeah, they did it in the validation studies
Yeah,
and
that was in the first
the
paper.
percentages between one scan, the other one. For the BTSP is like 6%, for the BT is
Yeah.
like 4%. So the numbers are not that far from
Right.
these differences.
It
Yeah.
would be really interesting here if you plotted to see for individual points, if there's a reduction in
Yes.
everyone.
Yes, yeah, to look at all the different,
And also,
yeah,
are these inpatient?
inpatient.
So maybe another analogy with your monkey study is that the
Social
individually housed, it's very stressful for
Well,
monkeys to be individually housed.
well, that's
It might be very stressful.
That was
for
actually something
people
that the
to be
reviewers, yes, in terms of the environment and whether the environment, initially the reviewers for the grant said, oh, but the environment is going to be different between the cocaine participants and the healthy controls.
Yeah.
So we said, sure, so we can do the study in the healthy controls as inpatient. And then they said, no, let's just keep it as outpatient.
Do you
Yeah.
measure cortisol levels and stress, indicators of stress in your?
No, we should, because there is it's a big, I mean, I think that just being impatient and is a big adjustment and stressor, and yeah.
So
Yeah.
does synaptic density, does it go down with healthy aging and with stress?
Aging changes it. In the initial study, we took age into account and we didn't see any.
Synaptic density goes down with age.
You speak in terms of- And I confirm.
This year you had to ask 5 people, last year was 4 people. Undergrads. But kind of just being that with people who are cocaine users, that their brain functions best on cocaine. If you are not on cocaine, whoop, you immediately have a stressful event. That's a very Dutch thing to
Frank
say.
Gavin and Herb
They
Cleaver,
try to
Mhm,
treat
who was
people.
studying cocaine use disorder at Yale
mhm,
40 years ago, 30 years ago, and it was all about the dopamine transporter and using discipramine to address the self-medication to augment dopamine availability. And they initially showed these wonderful findings, which were never replicated, unfortunately. So I think, we're still using the
Mhm.
same model of
mhm.
there being a neuronal deficiency of some sort. And I think it varies over time,
Mhm.
that people are self-medicating. And I'm just not sure that holds up. I mean, I just
Mhm.
think we see too many contradictory findings
Mhm.
for them.
But my point was because we're looking at all these patients after they've had this disease for a number of years.
Yeah.
So we're looking at we're looking at evolved phenotype. We're not looking at the baseline phenotype.
You're looking at the results, perhaps. I mean, that's
Right.
all. Chicken and
Mhm.
egg.
Yeah, so in terms of this is just to show that we didn't see changes in the centrum semivale or in the free fraction in terms of one scan versus the other scan.
Is this published?
No,
The first one?
the first one is, this one is where
Yeah. you're in
student starting it. So this opens the question for potential interventions, and in terms of interventions, and related with the question about the duration of the changes. So in Sterlis, they did one study administering ketamine to persons with major depression and PTSD. So they saw that in a subgroup of persons with the lowest synaptic density at baseline, when they had the baseline scan, ketamine infusion, and then another scan 24 hours later, in the subgroup with the lowest baseline, they saw ketamine having an effect, which one increase in synaptic density. We know that ketamine can do that preclinically. I'm also curious about some studies done at Columbia University examining one infusion of ketamine in cocaine use disorder and showing beneficial effects. So that could be a direction for future studies to see whether ketamine has synaptogenic properties that are beneficial in this group. And yeah. And psilocybin is, I mean, is the one that we're using for the opioid studies, but I know there is a study on psilocybin in Alabama, University of Alabama, and I think the Peter Hendricks, I think, yeah. So I'm curious about those results to see, yeah.
UAB.
Yeah, yeah. And
Is there any literature on any people or children with ADHD before and after stimulant?
so we wanted to do one study with ADHD, and the reviewers didn't like that we were not scanning minors. So in terms of one of the questions this morning
yeah.
about, so they say, no, you are not including minors. You should do it. And we said, well, it's because of the PET. I mean, it's not, I mean, we don't want to, the IRB part,
Adults. with
we
ADHD,
were proposing just adults. So,
which
yeah.
have a lot of exposure to stimulants, right?
Yeah, we
These
could-
are the same chicken and egg.
But you could still get a newly diagnosed adult with
Mhm.
ADHD.
Yeah.
You don't have to do, I mean.
Yeah.
But still, it'd be easier to find that than PET
Than
scan
doing
kids
kids,
with
yeah.
A lines.
But that's something that we could explore again with the NeuroExplorer, like
Right.
having higher resolution and the need for a lower dose. We will see if it is something that we pursue, but
Yeah.
nothing for now. Yeah. So I think that is, in a nutshell, all of this is thanks to the NIH, the Yale Pet Imaging Center, Bob Mallison, who was the mentor helping with these initial studies, and the CNIU. Yeah.
Great.
Yeah.
So. Question, now that you have the NeuroExplorer there, so you're gonna continue to do all your studies on the HRT for now.
Mhm.
But is there a benefit you think you could get out of the NeuroExplorer with this particular data set? Like,
Mhm.
one of the things we don't talk about is that the Explorer is a theoretically more sensitive machine.
Mhm.
And so that means, the data be more reproducible, or could you use a smaller group?
Yeah.
Because you have greater sensitivity to look at these differences. So is that something you
Yeah.
and Rich have talked about, or you're kind of just finish the study on the HRT?
Yeah.
Which is probably my mindset,
Yes.
and then
Yes.
go from there, see what you can do.
I mean, there is one idea which is in connection with the Yale Child Study Center. They had a cohort that they have been following for almost like 18 years. So it is a cohort of minors, now young adults, who were exposed to cocaine during pregnancy. So the idea is to test what is happening with them. And if they are like at 18 or 19 years old, age range, that could be an opportunity to use the NeuroExplorer and potentially lower doses, and to get a sense of endophenotypes and to what extent that exposure is still having an effect, or
I still think you probably still run into your chicken and egg.
Yeah.
With that group too,
Yeah,
because you don't know if it's the genetics
because
or the
it's portion,
development or
yeah.
whatever, but
Yeah.
what's the phenotype
Yes.
of someone who's not an addict?
Yes.
Is still, that would still be
Yeah.
helpful.
And in your cocaine group, did you test for fentanyl?
We are testing everyone
Yeah.
for, I mean, we see, but when we see fentanyl, it is just, I mean, we can tell them, I think that what you're using may have some fentanyl, but it's very, very minor amounts. Yeah. Yeah. We're now doing that for, I mean, for the opioid studies, we have not only fentanyl, but also salicin, and like more than 50% of the participants are showing salicin on the urines as well. Yeah.
So I haven't seen this problem.
Yeah.
I still love the idea of doing the xylazine
Yeah.
kappa study, see if it's
Yeah.
interacting with the
Yeah.
kappa receptor. Look for that. Thanks. Other question.
Yeah, but I think that the discussion about the environment, dominant, subordinate, I think that just seeing the participants on the inpatient unit, that is a big change for them, and maybe that environmental exposure could be having an effect on what we see.
The
three
other thing
weeks,
is,
yeah.
I assume that's their first PET scan with an A-line they ever receive. And so if you know what's coming, then your second PET scan with an A-line, you're gonna be not as worked up about it.
Mhm.
Yeah, less stressful,
Yeah.
Or
so
would you?
I don't know
Expect
about
an
that with the
immediate-
A-line. It may be more.
more stressful if you,
Oh.
depending how the A-line goes.
Yeah.
That's true. True. I'm sorry, what were you saying? Karen?
No. Well, yeah, would you really expect such drastic reductions just because of test retest? Yeah, it really seems
Yeah.
like an abstinence effect.
Mhm.
I agree. So are they using the drug to rescue their phenotype?
It would be interesting to maybe test them a year later or something when they're really recovered,
Yeah.
if they've really.
So much.
You know, those long-term studies are so appealing, but it's so hard to keep the variables under control when there's a long interval between, you know, the first and the second scan.
You know, like for example, for this one, we were very excited about the clinical component, like to see to what extent the time to relapse is associated with the PET, but almost like the most out of everyone had done some outpatient follow-up. Usually they do the PET and then they don't come back.
Right.
Yeah. Yeah.
Studies out there that just looked at a single dose, because then you can manipulate, of course, in a healthy volunteer, maybe not with cocaine, but with amphetamine.
Or a single dose of, yeah, we wanted to do that on the initial grant, like a hard exploratory aim on cocaine administration because Bob Madison had the IND for cocaine and
Or you can
they
give
didn't
cocaine
like,
to healthy volunteers.
no, on the cocaine
Oh, I
participants,
see,
yeah,
okay.
on cocaine participants, yeah.
That's, I mean, you can give to healthy volunteers.
Yeah.
right? So,
So if you're doing cocaine, you should contact Mike Nader and the group at Wake Forest. They're still doing cocaine self-administration. in one piece. You can control for
Yeah.
all of that.
Yeah.
In non-human primates, and those guys come back.
Did you? They don't go in
They
there.
have nowhere else. If you want to feel like a drug.
Cooler
effect,
for
Yeah.
bananas and cocaine.
Yeah.
Cocaine and the
They
rest.
still started the cocaine supplements, they did it one time, didn't they?
I mean, we kept the IND, but now we discontinued mainly because of all the, like interestingly, every time someone was, if they were coming to Yale to do an audit, and then they said, oh, let's just randomly look at one study. What do you have? They will always randomly choose the cocaine IND. Oof, yeah. It's like, I was
Don't
for so
worry,
many
next year
years
they'll show
ago.
your psilocybin
Yeah.
IND randomly.
Yeah,
every
but that
single
one I
time.
will be more comfortable with because it is like it's fresh. We're doing that
Oh,
work
yeah.
now, and
Those non-human
Yeah.
primates are really good for
Yeah.
looking at the effect
Yeah.
of drug on brain.
Yeah.
Yeah.
Right, because you control all the variables. Variables.
You use days of cocaine use. That's probably easier to quantify than a quantity frequency measure. How much do they use when they use? Since
Mhm.
cocaine is not susceptible to, Bureau of Weights and Measures,
Mhm.
the way alcohol is or
Mhm.
ATF. Because it's very hard to quantify.
Yeah.
What a person's using.
Yeah.
So I assume that's why you use days of use rather than a quantity free, because exposure is really what you're interested in.
Yeah. I mean, we have quantity as well. We're using that now.
What do you see? Is it better quantity frequency?
I mean, on this longitudinal study, we see actually that quantity is positively correlated with what we see with the first scan. So that is reassuring for the quantity, but we don't see that for the frequency of the cocaine use. So we see it for the dollar amount. We don't see it for the days of use.
So use dollar amount as a
Yeah.
metric.
Yeah. So that part is not making a lot of sense now. And we see those correlations for one PET outcome, not for the other one. So we'll have to,
But you're doing multiple comparisons.
Yeah.
On control.
Yes.
Are you looking at the 18F version, the Sinvest T? Or
Not on this one, mainly because hopefully down the road, one idea would be to combine this with the older cohort, but also to combine with the studies on alcohol, cannabis, opioids, so we can have a large comprehensive data set across substance use disorders.
Gotcha.
So because of that, we want to keep the same tracer and yeah.
But maybe a different scanner.
Yeah,
That's gonna be the question.
yes. You know, I think the HRRT, I think they will keep it for like maybe 2 more years.
Uh,
It's sort of retiring.
Very good.
Yeah,
Good
okay.
stuff.
Thanks a lot.
Thank you.
Thank you, Gustavo.
Thank you, Gustavo. We really appreciate it.